We were formerly known as the RTB Raleigh, North Carolina chapter, one of the local chapters of Reasons to Believe. Reasons to Believe retired their chapters program in June 2024. Welcome to our June 2024 meeting. For tonight's agenda, we have a medical presentation entitled The Human-Microbe Relationship by Francisco Paco Delgado, MD. And after his presentation, we'll talk a little bit about the new name and email address for our group. If anyone... All right, you know what they say on the other Zoom calls. If you object to being recorded, please disconnect now, but please don't. All right, so we're recording this. We will put this up afterwards. I'm sure I'll do a little bit of edits to it, but anyway, so we are recording now. So let me introduce our speaker. Some of you may know him. Here we go. Francisco Paco Delgado is a medical doctor with a specialty in infectious diseases. After earning his MD from Universidad La Salle in Mexico City and completing his infectious diseases fellowship at Vanderbilt University, he's been in clinical practice for over 25 years. Paco discovered reasons to believe in 1997 while researching different views of Genesis in the history of the Christian church. He's been involved with RTB Indianapolis chapter since its foundation and now serves as its chapter president. He lives in the Indianapolis area with his French-Canadian wife, Claude, and 3 daughters. Paco and his wife help run a cooking club and enjoy playing pickleball. Just as a side note, I had a chance to observe and play a minute of pickleball because I hadn't played it before. So that looks like a very interesting sport. So I'm going to stop my share. Paco, you can take over here.
All right. Can you hear me?
Yes, we can hear you. And I'm going to move your screen over to my big screen so everybody in the room can see you.
So how many people are in the meeting?
Let's see. I count. Well, here comes another one. I count 11 online. That, of course, that includes you and me. And we've got 2 others besides the 2 of us in the room here. And here comes Amy.
Perfect. It's amazing to see that, when we do our meetings here in Indianapolis, we get a lot of people just from around Indiana. Very rarely do we get people from other areas. I'm surprised to hear that you get people all the way from Denver. Is that Belinda, you're in Portland?
Yeah, I'm in Portland, Oregon.
Oh my goodness, that's amazing. One of the things about the chapters, I've been serving as a chapter president until since the beginning probably of the chapters, probably about 10 years or so. And it's amazing to see how much fellowship really the chapters gave. I felt that there's a lot of connection. So I'm very happy that you are continuing the chapter in a different way. We're trying to do the same here in Indianapolis. So anyways, it's a pleasure to be with you. I think that all these fellowships with people that are, that have this interest in apologetics, especially with science, is very special. So I am an infectious diseases physician, so I deal a lot with bugs and all kinds of microbes and everything. So we are going to be talking today about this human-microbe relationship. I gave a larger version of this talk at RTB. It is online. I've tried to update it a little bit, making it a little bit more general, and I am open to any kind of questions you may have about this topic, about anything in the infectious diseases area. I would be curious to see what you think about this and what is in your mind about how all of these microbes are function in the whole area of apologetics. Anyways, something happened. I didn't want to. There we go. So this is a little scheme of the human body. If you consider all of the cells of our human body, you may not know, or you may know, that most of the cells in the body are really erythrocytes, the red cells. Everything else is just a small portion. You've got, for example, here the epidermal cells are your skin. Muscle cells are very, very tiny. Some people tinier than others. bronchial cells, vascular cells, all of these are very small. The big bulk is really the red cells. But when you consider the number of cells, you got about 25 times 10 to the 12 red cells. Everything else is about 5 times 10 to the 12 number of cells. Bacterial cells that occupy or inhabit our body are about half, about twice, I would say, yeah, twice or about the same amount sometimes of the number of cells. So, and another important thing is, whereas this is the number of cells, then the amount of genes that bacteria have and that may impact our body is beyond anything you could imagine. Human genes are only probably about 23,000. Bacterial genes are about 2 million. We are in the very early stages of figuring out what is the impact that these genes, that the product of these genes or the modulation of these genes has in the human body. So a lot of what I will be sharing today is still unknown. In the last, I think it was about 7 years that I gave this talk, not much has changed. It is a very difficult area to research. We are getting new tools on how to deal with the research of this, but it is not easy. It is very, very difficult, just because it's hard to isolate some of these bacteria and then trying to understand what is the interaction between the bacterial genes and human body. It's also difficult to elucidate. Anyways, so if we see the number of humans of cells, human cells that you see here, the red cells, fat cells, other muscle cells, bacterial cells do not contribute much to our weight, to our mass. It's only a very, very small amount. And most of these bacteria inhabit the gut. There are bacteria all over our body, the gut, oral cavity. Interestingly enough, we thought at first that lungs were sterile, but now we know that there are bacteria that line the airways very deep in our lungs and that provide a protective layer. The skin, the urogenital tract, essentially all of our body is in a way inhabited by bacteria, especially those areas that are exposed to the environment. To the point that somebody said humans are superorganisms whose metabolism represents an amalgamation of microbial and human attributes. So these microorganisms play a very important part in our health. They are very important in our survival. And turn this off here. So somebody else said here, the gut microbiome is said to encode a second genome, and its functions expand the host's physiological potential. In the gut, microbes extend digestive capabilities. They prime the immune systems, produce vitamins, degrade xenobiotics, which is other products that enter our body, and they resist colonization by other pathogens, which are the bad bacteria, the bad microbes. In the gut, for example, the bacteria help us to metabolize food. They protect against other nasty bacteria that could give us diarrhea or other problems. They actually stimulate a healthy immune system, and they contribute to vitamin production. So without the bacteria in our gut, we would have really many problems. That's why when I see antimicrobial soaps that kills like 99% of germs, I go like, well, I'm not sure we really want that or we really need that. Now, this is an important concept that I'd like to introduce. The relationship between the human body and the microbiota is a very dynamic process. So let's say that today you went to the bathroom, you find this antimicrobial soap, you wash your hands with antimicrobial soap. and then you go out and about for the rest of your day. You may have killed most of the bacteria, let's say in your hands, but over a certain period of time, your body will start replenishing the bacteria to the point that your hands will be again covered with microbes after some time. How long it takes, it's a very difficult question to answer. It really depends a lot on the health of the person. If a person has let's say a healthy forearms, healthy arms, the bacteria are going to be more easily transmitted to the hands and colonized. And colonized means essentially that the bacteria will cover up again the old niches that were disrupted, in this case, by the antibacterial soap. So when you take, for example, an antibiotic, yes, your gut flora, your skin flora will be changed, but after some time, it will regenerate. It doesn't always regenerate in the same way that it was before the disruption. So this is a very dynamic thing. It changes, and if you take, for example, another antibiotic, or you wash your hands with antimicrobial soap, it'll change again. Bacteria will come back and recoat your hands, for example. The bacteria that may coat your hands, though, may not be always beneficial. So that's why when we disrupt the normal microbiome in our bodies, there is always a chance that what comes after could not be exactly the best bacteria that we need. We'll talk a little bit more about that. How do we get all these bacteria on our cells? We start essentially from the moment we are born. We still think right now that the placenta is a sterile organ. I wouldn't be surprised if we find some element of regulation that is going to be important for the adequate function of the placenta. I say this because we know that there have been genetic sequences of viruses that people have called endogenous retroviruses that make the placenta work adequately. Now, how did these endogenous retrovirus get in there? Not exactly sure. They are there, and they may contribute to essentially to a healthy development of the baby in utero. The mode of delivery then will give the baby a very special microbiota. Children that are born via vaginal delivery will be coated with something called lactobacilli, and children born through cesarean delivery will be coated by bacteria that usually live on the skin. Now, this is something that I've been looking closely. I'm trying to look for more studies that to see if these make any difference, because there seems to be some propensity for some diseases depending on the mode of delivery, but also on the mode of the infant feeding method. We know that breastfeeding gives the baby some microorganisms now from the skin of the mom, gives antibodies, and it also gives some cytokines, which are regulatory proteins that help train the immune system. Whereas if a baby is bottle fed, they are not exposed to these organisms. Again, how that makes a difference, not exactly sure. It seems that the breastfeeding and the vaginal delivery give the baby a better knowledge between what is a good bacteria and what is a bad bacteria. And then the immune system can learn to distinguish what to tolerate and what to attack. The way that I put it is like in this graph, if you in World War II, there were lots of airplanes flying around, and if you were a fighter pilot or somebody dealing with an anti-aircraft artillery, you need to know exactly how the friendlies look, how the foes look, so you don't shoot your own airplanes, your own people. So if you are in a situation where you really need to know what to do when you find something that could be threatening, we need to know exactly what kind of uh, of, of agent we're getting. We need to make sure that we don't kill what we're not supposed to kill. This is the function of the immune system. The immune system needs to distinguish between friend and foe and be able to eliminate the foe and tolerate and actually promote the survival of the friendlies. This is fascinating because now this is a very complex schematic. This actually is the diagram, and Dennis might know about this a little bit because you're in IT, but this is the diagram of a cell and its response to multiple stimuli. So the little ovals on the periphery are different stimulants. And every stimulation will trigger a completely different pathway in the cell, and it'll tell it exactly what products to produce or what proteins to produce, so they can either kill something or bypass something that is encountered through the human body. This is just one of the many cells in the immune system. So it really looks a lot like a motherboard. Yeah. So this is, when we talk about design, my goodness, I mean, each cell of the immune system, you can think about it as having its own motherboard with multiple pathways from input to output. Now, I am going to show you a little bit about how the microbiome interacts with the body using the skin as an example. In normal skin, you have bacteria, normal bacteria that are supposed to be there. We call it staph epi. This is a crazy name, but we just call it staph epi for short. Staph epi is supposed to live on our skin. It helps our skin. Staph epi is recognized by these cells called dendritic cells, which are cells of the immune system. And they pass that input, the information about their presence to other cells called regulatory T cells. So the dendritic cells are actually the sentinels, and the regulatory T cells are the command center for the immune system of the skin. So what happens is that staph epi will grow and it will produce these things called small chain fatty acids, which are essentially waste products. So when you have a lot of staph epi, there will be, of course, a lot of waste products. That means that there's an overgrowth of bacteria. So the regulatory T cells will recognize the excess of the small chain fatty acids, and they will then eliminate the excess of the bacteria, promoting back a normal number, a normal population of staph epi that is healthy both for the body and also for the staph epi, because too much staph epi essentially will end up killing itself. Staph epi also produces proteins that work as antimicrobials that will prevent the skin being attacked by pathogens. Staph epi, for example, prevents the appearance of Staph aureus. Staph aureus is a really bad. If you've heard about MRSA, that's essentially one strain of Staph aureus. Staph aureus can be very aggressive. And because we have Staph epi on our skin, we do not constantly get infections with this Staph aureus bacteria. But Staph epi also produces these other proteins called 6-hydroxyaminopurines that will kill tumor cells on your skin. So in a way, it is a cancer-preventing cell. Also, Staph epi will produce some other proteins that through the dendritic cells and other immune system cell called the cytotoxic T cells will help in the healing of wounds. So you can apply the same processes in other areas of the body that include the mouth, lungs, gut and vaginal area. Think about it. So the normal microbiome on your body will be preventing disease, will be preventing cancers in many of the areas of your body. But what happens then if the immune system is not adequately trained? Well, that's when you get friendly fire. You start destroying your own airplanes here. A bomber on the top is bombing a bomber underneath it. And it seems that there is a window of opportunity for this training to take place. This is another example. If we have, let's say, a colon, your gut, which is completely germ-free, these cells are other immune system cells called iNKT cells. And there have been some experiments in rats trying to figure out what happens when we expose these iNKT cells to different environments. So if the iNKT cells do not encounter any microbes, the patients will be susceptible to inflammation of the colon. It seems then that the cells will not recognize what is friend and what is foe, and will end up attacking a normal cell in the colon, causing inflammation, which essentially is what colitis means. Now, if we introduce during that window of opportunity, for example, here is in a mouse at the 2-week mark, if we introduce, and this is a key word, diverse bacteria. I put here these bacteria in many different colors just to indicate that these are diverse bacteria. Exposure of the cells to diverse bacteria will lead to a healthy colon. Now, if those healthy bacteria are present, but after the window of opportunity, we end up with inflammation of the colon. And if we expose these cells to just many bacteria, but of only one kind, not a diverse kind, only one kind, this will also lead to inflammation of the colon. So there is this window of opportunity at some point in the development of living beings that allows for a healthy outcome. So we have to, in human, this is all in mice. In humans, it seems that it is very similar, although the pathways are still very complex. But we do know that this, in a sense, does apply to human beings. Now, in the lung, same thing happens. If the cells are not exposed to pathogens, people will be susceptible to asthma, could be susceptible to asthma. A normal lung requires the same exposure to diverse bacteria before that window of opportunity. If those bacteria are introduced after, there will be susceptibility to asthma, and the same thing, just one bacteria at multiple levels will produce in that susceptibility to asthma. Same thing with the antibodies. There is something called anaphylaxis. Anaphylaxis is an overwhelming allergic reaction to something. You might have heard about people who have peanut allergy that may close their throat. And some people have died from severe reactions, let's say to peanuts, but there's many other things that could cause anaphylaxis. But the same thing happens if we show the immune system multiple bacteria during that window of opportunity. There will be a normal susceptibility to anaphylaxis, but in all of the other scenarios, there will be an exaggerated susceptibility to anaphylaxis. So it is now increasingly clear that early life colonization with bacteria also coincides with a potentially time-limited period during which the immune system is permissive to microbial instruction. So we need those initial microbes to teach the immune system how to differentiate between friend and foe. Now, this is what my representation of what a normal microbiome should look like. You have to have multiple bacteria, very different. That will give you an advantage, a healthy advantage. Let me see then, or let's consider what happens when we grow, let's say, in different environments. You have on the left, a rural environment, on the right, a more urban environment. This is a paper that recently came out, interestingly enough, showing how the exposure to microbes changes a lot depending on what kind of buildings we dwell in. If you have a hut, for example, which is very exposed to the environment, you will have air coming in, you may have exposure to animals, you will have exposure to dust, plants, pollen, whatever. And in our more, urban environments, you have these big buildings that have like filters and, air is filtered, everything is away from anything that would be probably called a natural air. It's a more clean air. And this kind of environment actually changes also the exposure and the development of a microbiome. And the last sentence here, impairing human exposure to nature and its element. And I think that word is very important, this word impairing the exposure, because that exposure is crucial for us to maintain our health. So in children that grow in a rural area, the Bacteroidetes are the good bacteria. These are bacteria that are very, very diverse, and they will give you a good gut microbiota. The Firmicutes are less diverse. They are more homogeneous in their function, in their biology. In an urban area, then you have children have more Firmicutes, less Bacteroidetes. So there's I would say a little handicap there for children that grow in an urban area compared to the rural area. We know that modernization, with everything it entails, we got now antimicrobial soaps, we got all these things that try to get rid of microbes. I see all these cutting surfaces that have like silver to eliminate all microbes, whatever. The more modernized we become, our diversity, the diversity of the microbiome decreases significantly. So we have seen, and this has been published, this is a paper from Science in 2016, modernization decreases significantly that diversity compared to communities that have minimal modernization. Now, where do we go from here is essentially where, it's up to us. How do we want to continue this graph? Do we want to continue the modernization, limiting exposure to microbes, and maybe impairing our health in different ways? Or do we want to change that and, be more in contact with nature, be more outside, spend less time with our iPhones? I don't know. Something like that might be a better idea. So things like water sanitation, less exposure to animals, less exposure to parasites, smaller families with fewer children, and antibiotics are some of those impacts that we know have made a difference in the microbiome of people. Antibiotics are a big thing right now. I gotta tell you, as an infectious diseases physician, most of my time with patient care it involves stopping antibiotics more than giving antibiotics. We spend probably about 60 to 70% of our time stopping antibiotics and only about 30% prescribing them. Antibiotic use within the first 6 months of life, in this paper, it says it's associated with an increased susceptibility to allergy and asthma at 6 years of age. There have been some papers that may be challenging this view, but the data still seems to be skewed towards, antibiotic exposure during the first years seems to have some issues with the development of an abnormal immune system. The second paper here, development of wheezing and eczema. Eczema is a condition of the skin in which there is the persistent inflammation. The issue with the asthma is a little bit clearer because we know that there's less asthma in third world countries as compared to first world countries. Exposure to antibiotics in childhood, especially during the first year of life, is associated with increased risk for development of something called inflammatory bowel disease. First year of life associated with increased susceptibility to being overweight, developing central adiposity, may lead to increased development of type 2 diabetes later in life. I wish I could tell you, this has been proven. No, again, it's very difficult to do research in these areas, and I was hoping again that I would find more definite findings after 7 years. But no, it's been essentially in the same place. There is a propensity, but it's not an absolute, oh yes, this is going to change your health in a certain way. Now, this is something very important. Overall, medically important antibiotics accounted for 62% of all antibiotics sold for use in food-producing animals in 2015, and 70% of all medically important antibiotics in the US are sold for use in animals. So even if we take care of ourselves, if the environment that we have, meaning here, farms, chicken, cows, anything, if they receive antibiotics, we are actually changing the environment in those farms, and they will end up giving us exposure to bacteria that are more pathogenic. We have seen it with multiple resistant bacteria that people come to the office or to the hospital with infections with multi-drug resistant organisms, and they say, but I haven't taken antibiotics ever. So we know that these are coming from, let's say, supermarkets, meats, chicken, whatever products are that from animals that have been exposed to multiple antibiotics. So we know that the human microbiome has an effect in all these multiple areas, obesity, inflammatory bowel disease. We know that there is a way in which it affects it. How much? I think it depends a lot on the person. I think it depends a lot in the genetics of each one of the people around. It's not like if you change your microbiome, you're going to have obesity for 100%. Some people will be propense to that. Some people will be propense to diabetes. Who are those people? Not exactly sure. Again, because the interaction between the bacteria and the human body is very complicated. But doing studies with cells, not with human beings and all, we have been able to find these things. Regarding obesity, antibiotics will make animals grow, and that is the reason why antibiotics are used in livestock. You give antibiotics to animals, and they'll weigh more. They'll essentially be selling for a lot more money than a lean animal. Now, animals that are given a single bacteria in high levels will also make them grow significantly. We know that if we give animals diverse bacteria, they will essentially have a normal weight, and animals that have high fat content will also have a normal weight. Now, how does that work? when we ingest healthy carbohydrates, in this case, fiber, for example, this is a very important example because I think it's something that seems to be more talked about right now in the literature. When you ingest good carbohydrates like fiber, these will be metabolized by the bacteria in a normal gut to chemicals that will help in the regulation of glucose and lipids, and they will essentially give you a normal weight. This part of the graph here says it's blocking this immune cell, this white cell, meaning it prevents inflammation, prevents inflammation of your body. In this case, it would be the gut. The products that the bacteria produce will be these fiber will be metabolized into something called carbohydrates that will go into the liver into a normal, I should call normal cycle. Whereas if you have abnormal bacteria, if you take, for example, sugars like sucrose, they will actually metabolize into different chemicals that will probably result in obesity, again, in those people who are genetically predisposed. So if you have normal bacteria, what you eat will translate into a healthy body, essentially. Now, those I would call bad bacteria or single bacteria or non-diverse bacteria can also produce other chemicals that will activate immune cells and will produce inflammation of the gut. This is what is thought to be some of the basis for what we call inflammatory bowel disease. Inflammatory bowel disease, people have a lot of diarrhea, have a lot of abdominal pain. Some people have constipation. The colon is very inflamed, and it is very difficult to stop that inflammation. We give steroids, we give other anti-inflammatories trying to decrease that inflammation so people can live normal lives, but it's a very ugly disease. Diabetes, some people with, again, the genetic predisposition, these bad bacteria will produce other chemicals that may also alter the metabolism in the liver and may then predispose towards diabetes. There's been a lot of talk right now also about promoting atherosclerosis. If you have the bad bacteria, it seems that remember the small chain fatty acids we talked about in the skin, these waste products will activate the immune system in a way in which there will be a formation of plaque, which are those accumulations of adipose tissue in the arteries, especially coronary arteries, and that can cause heart attacks. There is also a lot of talk about mental health, how a good diet will produce chemicals that will be healthy for the brain, but if you have a non-diverse flora, that non-diverse flora will produce other chemicals that may cause in some people mental issues such as depression and things like that. There seems to be that these could also be involved in some forms of Alzheimer's disease and in Parkinson's disease, but the factors that are underlying all these are not completely known. We have also other problems with say overuse of antibiotics. One of them is this bacteria called Clostridium difficile. Essentially, if you are colonized with Clostridium difficile and you take antibiotics, there's a chance that you can get very, very sick and could even end up in the hospital. Clostridium difficile has risen lately. It's a bacteria that probably was not very prevalent until about maybe 30, 40 years ago. in the last 40 years, we have seen an epidemic of C. diff, probably linked to the development of more powerful antibiotics. And interestingly enough, the treatment of C. diff uses other antibiotics. It uses something else called monoclonal antibodies. But we also use this thing called fecal microbiota transplant. So it may sound gross, but what we have done is we've been able to get this stool from normal people, and we introduce that stool in the colon of the health of people who have C. diff. We used to do enemas with normal fecal matter. Now there's a company that markets the capsules with normal fecal flora, and people take them. Anyways, it sounds gross, but I gotta tell you, it works. If we have patients with C. diff, this is a very good procedure to treat them with, which raises also the question of, if we use normal flora, can we, let's say, heal people who have ulcerative colitis or this inflammatory bowel disease? And in preliminary studies, it seems that yes, it might work. It might really help the people that that don't have a normal human microbial relationship. What was I going to say here? I'll remember. I was going to say something else here. Anyways, let me talk a little bit about probiotics. Probiotics is, many people come to me and say, hey, doc, can I take a probiotic if I'm taking antibiotics? And there's all these formulations out there that you can buy in the pharmacies. And my answer to them is this one. There is limited evidence for the role of probiotics as modulators of the human gut microbiota. Now, supplemental quantities of probiotics exert only a limited effect on the human gut. One of the problems that I see with the probiotics, or potential problems, is probiotics will give you a very high amount of very few species of bacteria. Meaning, remember the pictures I've been putting about the orange bacteria? They're all the same. There's a lot, but they're all the same. So that's what the probiotics really are. And that brings me a little bit of a concern, because then if you introduce those bacteria to a let's say a normal colon, then the colonization may not be the one that we want. This may allow then recolonization with different bacteria that could produce disease. There's also something about probiotics. Prebiotics are, I would call them bacterial food. They're defined as a substrate that is selectively utilized by host microorganisms, conferring a health benefit. So out there, and this is mostly in health stores, you'll find things like inulin, this thing called fructo oligosaccharides, lactulose, galacto-oligosaccharides. Essentially, I call them bacterial food. And that's an area where there's still a lot of unknowns, because some of them could have a benefit, some of them could be detrimental. And finding the right balance, it really requires, you have to play with the genetics of the person, you have to play with the history of the person, you have to see what is their makeup, so that we can give them the right bacterial food and the right bacteria, if you wanted to think about a probiotic, for them to restore a healthy microbiome. So it may be we may get to the point where we are going to be figuring out, oh yeah, this is the genetics of this person. We'll have a capsule with specific bacteria from healthy gut, and there's also special food in it that will restore a normal microbiota, let's say, in the gut of people. Anyways, there's a lot of unanswered questions in this area. One of them is, well, we've been talking about bacteria, but what about the viruses? I mean, there's so many more viruses than bacteria in our bodies. They all work also to regulate bacterial overgrowth. And the question is there any role for protozoa? those we still do not know much about, and I hope that at least in my lifetime we'll be able to learn a little bit more about what is the role of viruses in our in human health. I think that's going to be a fascinating finding once we get some answers. So my conclusions here are that the bacteria that constitute the normal human microbiota are specifically designed to maintain human health. The bacteria in our microbiota are established early in life. They play an important role in the training of our immune system. They influence many areas of human metabolism, and their disruption can lead to chronic diseases. And the benefit of probiotics and prebiotics is still unclear. Which it brings me back to, Psalm 139:14. I praise you because I am fearfully and wonderfully made. Your works are wonderful. I know that full well. I think when David wrote this, he didn't have the benefit of microscopes or all of the microbiology labs that we have. I don't know if if he could use any bigger words now to describe how all these things that he was unaware of are part of the good design and the good functioning of our human bodies. Anyway, so this is the end of my talk, and I will be happy to answer any questions that you may have.
Okay, either in the room. Yes, thank you very much. Either in the room or online, just unmute yourself
I can
and,
take out stop share. There we go.
okay, there we go.
Can you do it as a gallery? Dennis, right now we see you a lot. I think we don't want to see you a lot.
Yeah, I can do that. Let me change my speaker view here so we can see everybody.
There we are, equal opportunity.
There we go. All right. So in the room here where we are, let me get over here where you can see me. I've left the screen up there so we all can see everybody else now. So,
Okay.
all right. So who has a question? Anybody? Okay.
You talked about the difference between breastfed and bottle-fed infants. Does it matter if the bottle-fed infants are getting formula or getting breast milk in the bottle?
If you have breast milk, that'll be much better. From all the studies that we've seen, there is two things that have to be considered here. One is the exposure of the infant to the mom's skin. That's one, because that'll give the baby some bacteria that are on the skin of the mother. The other one is the components of breast milk. So there's two benefits by breastfeeding, but if the bottle with human breast milk with human milk should be actually better than formula.
Okay, great. Anybody else?
Amy has a question in chat.
Oh yeah, let me look at that. Okay, I don't know if you see the question in chat. What about best practices for hand washing then, based on what you said?
You go back to the Hebrews. They essentially wash their hands before eating. That is really the best thing. That's the most important thing. If we're going to be, thinking about the medical field, just washing before eating will prevent any soiling from pathogens that are overexpressed, let's say, if you on the skin to get into your body, which they're really not supposed to go. At one point, remember I talked about this dynamic mechanism. How do you get sick? Let's say that when you have the wrong bacteria in the wrong place at the wrong time. So if and probably it goes back to saying, everything could be detrimental at some point. Let me see. If you ingest one bacteria that is a salmonella, what will happen is your body will get rid of it. You'll never knew that you were exposed to salmonella. If you ingest 1 million bacteria that are salmonella, then the chances for you getting sick are going to be much higher. What hand washing will do is, if you have been exposed to an abnormal amount of bacteria. It'll decrease the load of everything. So washing it actually has two things. One is the mechanical function of the water. It's always better to wash with running water because that mechanical action actually will get rid of things like C. diff. Soaps do not kill C. diff, the C. difficile bacteria that I show you about. Soaps do not kill it. It's the mechanical running water that will get rid of most of the spores. And the soap will then decrease the bacterial loads of all bacteria, which is perfectly fine. I mean, if you're a healthy person, then your body will replenish them in no time. It'll be very quick. I don't know if that helps. I hope it answers the question.
Okay, great.
I would assume that also hand washing before preparing food would be recommended.
Absolutely. Absolutely. I mean, it's all common sense. You just have to think about decreasing bacterial load of something that would be pretty bad. So yeah, if you're gonna, it's not, you know, I put only that example, but yeah, preparing food, if we are going to be dealing, you know, in surgery, for example, yeah, we have to wash our hands to prevent the introduction of bacteria into areas that are not supposed to have any bacteria at all. So anything that is potentially disruptive to the introduction of bacteria in a healthy human being.
Okay, anybody else?
I had a question about probiotics, which you sort of answered. But given what you have told us about the limited evidence and so forth, if we take the position, it's kind of like a vitamin, it probably won't hurt, even if it doesn't help as much as is claimed. Should we be taking different brands of probiotics, switching off with every bottle to get exposure to different strains of bacteria?
You know,
Or
what
is
I tell my patients when they ask me about if I should take a probiotic, I tell them, listen, you like yogurt? Take yogurt. It's cheaper, more nutritious, and it'll give you exposure to more of the more bacteria, a more diverse bacteria than a probiotic will. So you could think about taking the different probiotics. It's just going to be more expensive and may not help you. Now,
Okay.
we have seen, for example, instances in which people who have impaired immune systems that take probiotics actually will get infection from the bacteria that are in the probiotics.
And I'm guessing, just to follow up on Loretta's question, if you look at the labels of all these probiotic things, will they tell you, oh, here's this bacteria, this one, and this other brand has these other ones, or can you even tell?
They'll tell you, but again, a healthy microbiome has to have thousands of different ones, and the probiotics will only have like 3, 4, 5 species. Yeah, so they're not very diverse.
Which is
That's why the fecal microbiota works much better for C. diff than anything else, because when you give normal, you know, stool to someone, that's thousands of different bacteria.
right. The North Carolina State Veterinary School does an open house once a year, and they have cattle there that are like fecal donors. They have them with a permanent surgery that it's sterile for that animal somehow. They've got some sort of protection over it, but they can reach in with gloves and get a sample and give it to an animal that is ailing, and it will cure that animal, which is gross and fascinating all at the same time. The idea of packaged probiotics, for lack of a better word, not having sufficient variety, is that another reason to seek out variety in the foods that we eat? Because
Yes.
they're going to have been exposed to different
Yes.
bacteria along the way. Okay.
One of the other things that I tell the patients that come in sick with the C. diff is just resume your regular diet as soon as possible. Just eat all kinds of foods, because yes, that'll help a lot more. Yes.
Interesting. Dennis, I see a second hand up or chat or.
Oh, let's see, I gotta come around here where I can see.
It just says chat.
There we go.
That's me.
Oh, okay.
I see Steve's hand up. Go ahead, Steve.
Yeah, I had a question, Dr. Delgado. It was an interesting presentation. I had a situation where I was very young as an infant and I was given penicillin and sulfa, which are two different classes of antibiotics. I had a reaction of some sort, small one. And for 50 plus years, I had to always say I might have an allergy to penicillin or sulfa until I went to the allergy lab a year or two ago and found out I was, in fact, allergic to neither of them. And now I'm left with a possible allergy to Cipro. But so my question is, I had read a while back that drug companies were really not working on new antibiotics. They were working on things like Viagra and, stuff like that where they can make lots of money. And I wondered if that situation is still the same, where there's potentially, with increasing antibiotic resistance, there's this situation being addressed now where they are developing antibiotics potentially to combat this problem.
Yeah, we do have new antibiotics, but they're few and far between. And the reasons are, yeah, as you say, the remuneration for antibiotics is very low for the pharmaceutical companies. We do have some, and what we're trying to do right now in all of the hospitals is whenever a patient comes in with bacteria that are very resistant, the use of these new antibiotics are restricted. It's restricted only to the infectious diseases physicians. So we are the ones that will definitely decide whether these people need to be exposed to these ones or not, trying to save them from overuse. And that way, we don't want to run out of options for patients. Yes.
Okay, Martha.
Well, it's interesting that Steve just mentioned about, I had a reaction to penicillin as a child as well. And I've often wondered, is that a reaction or an allergic, an allergy? And we've just never tested it.
Well,
We
it's
have a way
interesting,
to test.
Martha, what I found out in the reading and the doctors have told me that a lot of people my age were told because they had a slight reaction or a rash or something that, you're allergic to penicillin. They threw up their eyes, you can't take penicillin. And what they're finding is that, in fact, most of us are not allergic to penicillin, never were, and that we have a sensitivity to these things, but we're not allergic to them. So we've got two different things happen. Sensitivity, you get a rash and things, but you're not actually allergic. And specifically with penicillin in my age group, they were finding, a lot of people that were told they were allergic to penicillin really never were and can take it safely without any problem.
We have a test in which we can tell if people are allergic to penicillin or not. Not all of the hospitals have it, but when we have it, it helps us a lot because then we can differentiate those who are really allergic to those who, they really are not allergic at all.
Right. Well, my other question, I just found that interesting that I had that same situation, but are there any, I was a number of years ago, I was hospitalized with MRSA and had a number of operations and such. And I was on vancomycin for probably almost 6 weeks. Are there any long-term problems with that I should look forward to?
Yeah, it's a good question, but we don't know the long-term effects. Probably, and this is the thing, I think that the biggest problem would be in a young person, a child. I don't think that an adult will have much of a problem with the long exposure with antibiotics, except for bacterial resistance. That would be the main issue. But you go back and thinking, well, this is a very dynamic process. So once the vancomycin has washed off your body, and if you're living a normal lifestyle, you're exposed to now your normal things, normal foods and everything, most likely the bacteria in your body are going to change back to the regular ones. You see, what happens with bacteria, this is back to economics. All these resistant bacteria, they have to invest a lot of their energetic resources to keep the antibiotic resistance, which makes them less fit to compete against a bacteria that doesn't need to be carrying that load. So that's why if you let things go over time, usually these revert back to normal in an adult.
Another thing that I had been told, and this was back in 2007, and the last time I had any sort of a flare-up was in 2012. And it was right before I was very stressed. It was right before I was supposed to have my first knee replacement surgery. I mean, the week of,
Yeah.
bad timing, right? But they went ahead with the surgery and said, I think it's really okay. And so I haven't had anything since then, but I was always told to be very careful. And I've got, I always mispronounce it, muciprocin or mucipro. Anyway,
Muciricin.
yeah, and I, for my nose, because that's where it's harbored. And am I a danger? I've always been told that, it's a
No,
possibility, but it's been so long ago now that I've never had another flare up that I think I'm not a danger.
correct. So remember, this is again a dynamic process. You could be colonized with this MRSA at one point, but then your body will get rid of it. And then you may pick it up and and you never know when these events happen.
Right.
So that's why when I say, when you get an infection, it's because you have the wrong bacteria, the wrong place at the wrong time. So it may be that, the bacteria are gone. Can you test it? The problem is if we, and many patients ask me this, if I test you right now for MRSA and I get a negative test, I'll say, yeah, you don't have it. But I have to say, you don't have it right now.
Yeah. Yeah.
Tomorrow, I don't know.
Right. Yeah. So, I was like, it's interesting that you said sort of the vulnerability of the MRSA or the staph is that it's most of its resources are taken up with being resistant. So I guess that's its vulnerability as well. So, yeah, I was very, very cautious and very careful. Even now, and maybe I shouldn't, but I was recommended that I shower with Hibiclens, once or twice a week, but add that to myself. Maybe I should stop doing that. I don't know.
Again, being a very dynamic process, the Hibiclens is a very, it kills a lot of different bacteria. It's like using an antibacterial soap.
Mhm.
Many people develop reactions to the Hibiclens as well because it's a very aggressive chemical.
Yeah.
So I don't know. I don't think that I haven't seen any data on chronic use of Hibiclens to prevent infection with MRSA. I have not seen that.
Mhm.
I would say, and some people use it, which is, if you're going to have, let's say, a surgery, cardiac surgery, or a knee surgery, knee implant, hip implant. Yeah, we use the Hibiclens to try and decrease the chances that MRSA could be lurking on the day that you are going to be having the surgery. But after that, I don't know if there's any benefit on that, to tell you the truth.
Well, and I quite frankly, the past few years, in fact, I just noticed it was still in my shower stall, and I was like, well, let me see. The pump didn't work, so I was like, okay, well, I'm not going to use that right now anyway. So that brings me to my last question. Hand sanitizer.
Yeah.
There for a while. I was gun shy for a long
Yes.
time, and I'm still pretty gun shy because
Yeah.
I know there's bugs out there that can hurt you real bad.
Yeah. So, again, we had at one point, probably about 10 years ago, 5 years ago, we had an epidemic of this MRSA thing. MRSA is a very nasty bacteria. There were the strain that we saw the epidemic with. You see, the original MRSA had about 50 genes, let's call it. It was more than like 30 genes for bacterial resistance. They was resistant to multiple different antibiotics, okay? But then there came this strain that had this specific plasmid that would produce the resistance to the usual antibiotics, but it also carried about 33 different poisons with it. That's the one that we saw the epidemic with. And then we would, these bacteria were very difficult to get rid of. And that's when we encourage people to, whenever they would be in contact with, other people at other places, to use the sanitizer. Now that the epidemic is gone, we're not recommending people to use it that often. There are some people who, again, genetics, they are colonized with MRSA, and they keep developing infections with MRSA. And for those, Yeah, I could see the reasoning behind saying, yeah, just be more careful. But this is a very individualized advice. Yeah.
Yeah, it's interesting. When I got it in 2007, I'd never heard of it before, and it was not, you didn't really hear of it that much. And then soon after that, or within a couple of years of that, you heard about it all the time. So it was new to me. I was
Yes.
not glad to be a guinea pig.
I have a food question. Actually, this comes from Deanne. Oh, Belinda, do you have a question?
Oh, yeah, I do. Let's see if
Okay,
I
go.
can.
Yeah.
It's a, so with these conclusions, and it's more of a human origin question in a sense. So, it's that the bacteria is very important. There's a whole bunch of them that we need, and that they're involved in like our immune system, influencing our human metabolism, and such. So I just wonder if you have an opinion about that when God created Adam and Eve, that they had bacteria, a variety of bacteria already present. And then the other part of that question is bacteria are constantly replicating and dying and such. So there's that process. So that would mean that there would be death before the fall. So what are your thoughts about that?
Absolutely. Second question, absolutely. There's going back to Genesis. Bacteria are it's like, when did God create bacteria? Well, I would have to say when he created anything that lives, the very first cell, the very first organism, whether it was a cyanobacteria or something, there had to be, first of all, viruses. And there also, at that point, the introduction of bacteria may be at the same time or similar or close to when plants appeared. The biology of bacteria and the biology of plants, in a way, it's not the same, but kind of similar. So I think when God created Adam and Eve, it's like giving birth. They must have been colonized right away with whatever was in their environment. And that, I think, then helped finalize the immune system function. I think that's probably the way it might have happened.
Okay, thanks. I was, as you were talking, I was thinking about that, and then interesting to maybe be able to start conversations about, if they were there, then they had to die, or else we would be overrun with bacteria and viruses.
Absolutely.
Yeah. Thanks.
Great question, Belinda. So the question I have is relating to food. Is there any food that we should just generally avoid? And is there any food that we should definitely all be eating?
So in the papers that have talked about diet and the promotion of bacteria, what is a good bacterial food, what is a bad bacterial food? Basically, processed foods will have chemicals that will promote the growth of the bad bacteria. You go back to eat your fruits and vegetables and your fiber, and you'll be fine. But anything that is processed has the potential of having chemicals that will promote the growth of the bad bacteria. It's like it's so funny because it goes back to common sense. Just eat natural food.
Okay, great.
Dr. Delgado, the definition of processed food, I see
Yeah,
that.
I know.
Does that
Yeah,
mean
so.
anything other than fruit and meat?
Yeah, so
Processed?
meat is not a problem. The
Okay.
processed foods, anything that is in a package that has preservatives, anything that has been treated with oils, with fats that are not from necessarily and quote unquote natural origin may be a problem. Yeah, basically.
I mean, is that like pretty much everything in the grocery store, actually?
Packaged, yeah, mostly, yeah.
Okay, so it's eat natural, is what you're saying.
Yeah, yeah. Yeah, as much as you can. And again, that's why it's so complex, because not everyone will develop these problems even if they eat just, packaged food or processed foods. But I think as the human body has a lot of buffering mechanisms. We can try to kill it with many different ways, and our bodies, produce so many things to say, no, no, wait a second, let's get rid of this because it's not good for you. So it's not a all or none response. I think there's a lot of nuance from person to person.
Amy has a good question in the chat, which I will read here. Her comment is, I guess the death of a single-celled organism seems like it's a bit different than the death of a larger organism that has the breath of life. Do you comment on that?
Now you're getting into theological questions
Yeah,
here.
right. It's theological, philosophical at this point. Yes. Great question, Amy.
Yeah, I think that the theology here is the breath of life really means, the spirit of God. Is that what we mean? I mean, I mean, I think that's a whole different topic. But I would say that, people, I think one thing that I see, for example, in this debate about abortion and all that stuff, people say, well, how do you know it's alive? And it says, and I've heard this response a lot. They say, oh, that's above my pay grade. I'm as a physician in the hospital sometimes, not lately, but when I was in residency or anything, I was always asked to come and pronounce people dead. Well, I need to know if they're alive to know if they really are dead. And it goes back to the abortion thing, saying, well, yeah, of course, if you have a one-celled embryo, one cell in the human body that is replicating, we know it's alive. A bacteria is alive. It has all of the signatures that we consider life. So a bacteria dies, oh yeah, a bacteria can die, and they will die very frequently. The same thing, if we know an embryo that starts replicating, yes, it can die. Many of them die for multiple reasons, even spontaneously. We don't know it, but we shouldn't be killing them. We shouldn't be killing those embryos just because, oh yeah, we don't know if it's alive or not. Well, we know it's alive. Anyways, I got into a real rabbit trail there.
Could I ask a question? Thank you very much for your presentation. I really appreciated it and learned quite a bit. I have a question about, well, let's go back. Typhoid Mary.
Yeah.
I heard Typhoid Mary was a carrier, and I wonder if you can say anything about your experiences with carriers or what that involves.
Yes. Okay. So, a carrier is someone who essentially has a bacteria or I suppose it's usually a bacteria or a parasite. You can have it in your body. It may not be causing disease in yourself, but because that organism replicates in your body, you can shed it. Multiple ways to shed it, stool is the most common one. And that same organism can go to someone else and cause disease. So a carrier then is someone who has a bad, we call a pathogenic, an organism that can cause disease, but doesn't cause it in you, but can cause it in someone else. Yes. So there's a lot of people, for example, that like Ms. Potter Martha was saying, many people are carriers of MRSA. The MRSA doesn't do anything to them, but then they can pass it along to other people who are vulnerable and can develop disease. So yeah, that's the concept of carrier. Does that answer the question?
Yes, that answers my question. Thank you.
So I have a follow-up to that question then. So what is it about the carrier that they don't get the disease And can we bottle up whatever that is and give it to everybody else?
That's a great yeah. You know, it goes back again to the genetics of the person. Not everyone who is exposed to MRSA is going to develop disease. Why? Don't know. Is it because is it because of their own genetics? Is it because they have bacteria on their skin or on their nose that will prevent this Staph aureus to establish a home in there? Or there's an interaction between both that is actually a healthy interaction that makes a block against the MRSA. I don't know. Yeah, but there's a lot of factors in there. Yes.
Okay, great. Anybody else?
I have a question. First of all, thank you. A very informative talk. Um, in looking at this in terms of design, it seems like it's very important for bacteria to inhabit various places on our body. And you alluded to the fact that you have to have viruses to control the bacteria and so forth. So it seems to be a number of design things that potentially could be in play. Is there any way of quantifying those values to, I mean, I could see somebody who came from a naturalistic evolutionary perspective saying, well, these things just kind of grew together and that's how we have it now. But is there a way of, I know in biology it's a lot different than it is in some of the physical sciences and getting numbers, but do you have any feel about the quantification or the way that you could present your argument in a way that would be maybe more persuasive to somebody who maybe had a different view?
That's a great question. What is your background?
I'm a dermatologist.
Oh, there you go. That's a great question. I know, and you being a dermatologist, you know the biology is very difficult sometimes to quantify, especially when we don't know what parameters to quantify. That's probably the other issue. Yeah, the challenge here is, people say, oh, this was coevolution. Well, okay, coevolution. How do we know that these were the right bacteria? Well, because if they were right, people would live. If it was wrong, people would die. The relationships and the interactions are just so complex. I don't even know. I don't even think we have started to scratch the surface of those interactions. I think that it's going to become like, when Darwin wrote his book, it was like the protoplasm of the cell. We don't know what's in there, but it's working. I think probably, 100 years from now, we're going to go and discover that all this interaction is like now what we know now about the cell. It's so complex and so incredibly fascinating that, we'll still find signature of design in all these areas. Yeah, I don't know. If there's a way to quantify it right now, I wouldn't know how to do it.
I wonder if this runs into the problem of not enough time to try every permutation that would be required.
Probably. But I don't know. I really don't know.
I don't know if this, I listened to Discovery Institute quite a bit, and they approach the argument with that there's a mind. Well, we do as well. But I even think that Rana did this as well, where there's like forethought, like in this design that we're seeing in the cell, that there's manifestations of engineering kind of principles and those other type of principles that are that can only really be generated by a mind in a sense. So at least that's what I'm hearing from the Discovery Institute and using biology. So they're kind of coming up with a theory through biology of design, and that's using some principles that a mind uses to create, and that we see that in these bacteria and the cohabitation in a sense
Yeah.
of it as
Remember that,
well.
that scheme I put with the motherboard and all the activation of the T cells? That's probably what we're gonna be finding, all that the interactions will be between the human body and the bacterial genomes. So, but still, that's a very, very black box at this point.
Yeah.
All right. I do have another topic to bring up, but unrelated to your presentation, but any other last questions before we switch gears here? All right, well, thank you. Thank you very much. I'm gonna,
Thank you very much.
yes, thank
so
you.
much. That was-
Yes, I think we can apply for that one. Right?
Yeah.
Oh, thank you
Thank
so
you.
much.
All right, I'm gonna switch gears. I need to grab my screen back off the big screen and bring it back over here so I can see what's going on. All right, great. All right, I have an announcement. This is the new name of our group. We are going to be called the Triangle Apologetics Community. All right, and there's an email address that you see there. So those of you who are not on the email address or on our email list directly, if you want to be on it, copy that down and send an email to there to say, hey, put me on the list. Everyone who's currently on the RTB Raleigh chapter email list will receive an email tonight. And by tonight, I mean in the next probably less than 5 minutes, because I scheduled it to go out at 9 o'clock, welcoming you to the new list and asking you to do a reply all to that email if you want to stay on the list. And I'll let that run for a while before I start deleting ones off the list who don't reply. So be sure and reply all to that list. When you do reply all, it'll reply both to the current RTB Raleigh email address and the new one. As long as I get it in one of them, that's all I need. But that's what's going to happen. So in probably within the next 5 minutes, everybody who's on the list will get an email.